This is the story of a procedure that has become the single most common operation with over 35 million performed worldwide by 2010. It has evolved into a 10-minute process that can correct 96% of all refractive errors with minimal discomfort, a recovery time of a few hours, and dramatic visual results overnight.
Yeah, I can see the clock.
It is the confluence of numerous brilliant ideas and bioengineering accomplishments that have led to what is one of the most miraculous medical procedures in the history of medicine. The procedure is called laser in situ keratomileusis and is commonly referred to as Lasik. The concept that refractive error could be corrected by sculpting corneal stromal tissue to change corneal curvature was the brainchild of José Ignacio Barraquer Moner in 1948. Barraquer developed a procedure he coined as keratomileusis, which involved resecting a disc of anterior corneal tissue, which was then frozen in liquid nitrogen, placed on a modified watchmaker's lathe, and milled to change corneal curvature. The word keratomileusis literally means sculpting of the cornea. The resection was achieved by a manually driven microkeratome that he designed specifically for this purpose based on a carpenter's plane. Barraquer then used trigonometric calculations to derive the volume of tissue removal required for a particular refractive error correction. In his 1964 thesis on the law of thicknesses, he described that the cornea flattens when tissue is removed from the center and steepens when tissue is removed from the periphery. His earliest patients were treated in the early 1960s at the Clínica de Marley in Bogotá, where he had to leave the patient on the operating table after resecting the corneal disc while he hurried 3 kilometers across town to where he had set up the lathing workshop in his home, before returning to suture the thawed and reshaped corneal disc back onto the patient's eye by a torque anti-torque suture, using microsurgical instruments and an operating microscope, all of which were inventions of his for this purpose. Around that time, others were experimenting with Barraquer's ideas. Krawitz in Poland published a 1964 paper describing a series of 3 highly myopic eyes in which he had performed a stromectomy, where he manually made 2 stromal cuts at different depths with a flat knife and removed the thin lamella of intervening stroma. And Pureskin in Russia described the concept of an incomplete anterior corneal resection in order to leave a naturally hinged flap. Several thousand keratomileusis procedures were performed at the Instituto Barraquer de América in the 1970s and early to mid-1980s, and surgeons from around the world came to learn this difficult but miraculous technique. 2 of Barraquer's disciples worked on refinement of the technique to perform keratomileusis without freezing, referred to as the Barraquer-Crumaich Swinger technique, and this was published in 1986. This BKS non-freeze technique involved placing the resected disc epithelial side down onto a curved suction die or mold, where a second pass of the microkeratome removed tissue from the exposed posterior stromal surface according to the shape of the die. The BKS technique aimed to reduce surgical trauma to the tissues and improve visual recovery time. At around the same time, another non-freeze technique called in situ keratomileusis was developed. The procedure was first performed by Ruiz, who, having completed his residency at the Barraquer Institute, was performing up to 20 keratomileusis procedures a day. Ruiz was interrupted in his flow by a corneal disc resection which was found to be too thin for the required stromal tissue removal. With the patient on the table, he came up with the idea of passing the microkeratome a second time using a different suction ring with the height adjusted to resect the required lenticule directly from the stromal bed. This was called in situ keratomileusis. Ruiz was then responsible for designing a gear system to automate the passage of the microkeratome head. This eased the technical challenges of using a manual microkeratome, as the head could be passed with a constant and reproducible speed, therefore avoiding irregular resections and greatly improving the accuracy. The procedure became known as automated lamellar keratoplasty. Commonly known as ALK, this procedure was further refined by replacing the disc without a suture, and adhesion was aided by drying, after which the eye was patched overnight until the epithelium sealed it into place. In 1989, Ruiz presented a paper demonstrating how a flap could be produced by stopping the microkeratome before the end of the pass. The flap would then be tucked under the second microkeratome ring applied for the stromal resection, thus leaving a hinge to simplify the replacement and reduce cap-related complications. In the early 1990s, in situ keratomileusis was combined with the emerging technology of excimer lasers for corneal tissue ablation to finally become laser in situ keratomileusis, with the birth of Lasik as we know it today. But the excimer laser has a history of its own, starting in 1917. After Max Planck described the quantum theory, Albert Einstein predicted that the energy released by an electron moving from an outer orbital to a lower energy inner orbital could be initiated by an external source, which he called stimulated emission. It was not until 1952 that Einstein's theory was made a reality when microwaves were used as an external source to produce microwave amplification of simulated emission of radiation, or a maser. Later, the microwave was replaced by light, and maser became laser, as first described by Gordon Gould in 1957. The breakthrough ideas were the use of optical pumping to kickstart the process and to enable amplification of the emission by using a pair of parallel semi-reflective mirrors to partially reflect photons back and forth through the active medium. In 1970, the term excimer laser was introduced by Bazov to describe a laser built using a xenon dimer gas, the name excimer coming from an abbreviation of excited dimer. It was not until 1981 that an argon fluoride excimer laser was fired onto organic tissue when researchers at IBM made an incision in the leftover cartilage of a Thanksgiving dinner and found no evidence of damage to the surrounding tissue, unlike the charring seen around an incision made with a solid-state laser with a wavelength of 532 nanometers. They demonstrated that complex patterns could be made at a micronic level with each pulse removing a fraction of a micron. This research culminated in excimer lasers being used for etching microchips. At the same time, Taboda also found no thermal damage to the remaining tissue after a 248 nanometer excimer laser pulse onto corneal epithelium. Thus it was established that excimer laser tissue interaction was effectively non-thermal, but rather one of direct splitting of molecular bonds with minimum adjacent heating. This process was coined in the term photoablation. Having seen Taboda's work, Troquel contacted Srinivasan at IBM in order to investigate the potential of using their excimer laser to improve the accuracy of radial keratotomy incisions. Troquel later began working with Marshall to study the ultrastructural aspects of corneal photoablation. They compared the quality of the wounds made by an excimer laser at 193 nanometers with one at 248 nanometers, as well as made by a steel and diamond blade. The quality of the wounds was best with 193 nanometers. This finding was in agreement with similar studies by other groups at that time. The wound quality suggested to Marshall that large area ablation could be performed in the central cornea rather than just for peripheral linear incisions. This was described as photorefractive keratectomy. For PRK to become a feasible procedure in human eyes, the following criteria needed to be satisfied. First, the depth of tissue removal required for a given refraction change must be known. Munrelin proposed an algorithm based from Barraquer's earlier formulae to calculate the ablation profile as a function of refractive error and optical zone diameter. Second, the quality and clarity of the ablated surface must be preserved. Earlier studies in rabbit corneas demonstrated only limited haze after large area ablation. Myopic ablation on a donor eye also showed that the ablated surface was clean and smooth. Third, the wound healing process must not result in scarring. It was already known that there is no scarring after radial incisions. Then Marshall demonstrated no changes in corneal transparency 8 months after PRK in 12 monkey corneas, and MacDonald reported stable dioptric change in a primate cornea with good healing and long-term corneal clarity up to 1 year after PRK. In 1985, Seiler performed the first large area ablation to a human eye to remove a corneal scar. With the increasing interest in the possibility of performing refractive correction with an excimer laser, the first international workshop on laser corneal surgery was held in Berlin in 1986. This led to PRK being performed in humans. In early 1988, MacDonald performed the first PRK on a sighted eye due for enucleation, while around the same time, L'Espérance and Seiler began also performing PRK, but in blind eyes. Central ablation of the cornea was performed using different methods. L'Espérance suggested small scanning spot excimer lasers, which could be controlled to ablate in specific patterns. Marshall proposed using a broad beam laser and a moving iris diaphragm to shape the area to be ablated. For a myopic correction, initially the aperture was sequentially reduced in diameter to create steps in the corneal surface. The number of steps was gradually increased to improve the smoothness of the curved surface. Hanna used a rotating slit laser delivery system. The shape of the slit was determined mathematically to obtain a parabolic ablation profile that resulted from the slit rotation. Larger clinical trials followed with commercially available lasers given the encouraging results from the first cases. In 1991, MacDonald reported that myopic PRK treatment with the Vizix 2020 system was safe. In the same year, Lindstrom demonstrated the safety and efficacy of the Taunton Technologies Model LV2000 excimer laser for myopic PRK. At that time, Gartrey and Seiler also published outcomes using the Summit Technology Eximed UV200 excimer laser. Also in 1991, Dausch and Schröder presented results in high myopes with the Aesculap Meditech excimer laser, and later in 1993 presented the first hyperopic ablation profiles. At this point, the excimer laser story joins the keratomileusis story to become laser in situ keratomileusis, or Lasik. The idea of using an excimer laser to ablate tissue under a flap was springing up independently in many parts of the world. In 1988, Rajev and co-workers in Novosibirsk began using a 5mm trephine to produce a central 100-micron deep circular keratotomy, and then a scalpel to create a lamellar hinged flap. They then used an excimer laser to ablate the stromal bed before replacing the lamellar flap in 4 myopic and 5 hyperopic eyes, and presented their results with up to 2 years follow-up in September of 1990 at Columbia University. At around the same time, Burato was performing classical keratomileusis, but from October 1989, he used the excimer laser for ablation on the underside of the cap and published his first 30 highly myopic eyes with few complications and 1 year follow-up in 1992. Palikaris also independently conceived of a hinged flap using a microkeratome he had specifically designed for rabbit studies, and performed the ablation with an excimer laser on the exposed bed, followed by replacement of the flap without sutures, and first used the term Lasik in his 1990 paper. Palikaris treated his first patients in October 1990 and published his results on 10 high myopic human eyes with 1 year follow-up in 1994. It is this technique which gave birth to what is now the most commonly performed surgical procedure in the world and realized the dreams of José Ignacio Barraquer Moner 40 years in the making.